Welcome to the Everything Epigenetics podcast, Dr. Katz. I'm excited to chat with you today.
Thank you for having me.
Yeah, so you have a really interesting story, as we were just chatting before we came on air, that I'm super excited to hear. So, you're a classically trained developmental geneticist, and your interest really lies in, at first, I guess, in the development of germline of the roundworm, C. elegans. So, I want you to go into this story. I want you to maybe, you can even start with the end if you want to, tell people your greatest discovery and then how we got there. And we'll definitely go in and explain, what your evidence behind this particular finding is as well.
Yeah. So, my lab actually set out to understand how histomethylation can sort of function as information and can potentially be inherited cell to cell and even from generation to generation. And we were doing this in C. elegans, as you pointed out. And the end of the story is we think that Alzheimer's disease is actually an epigenetic disease in which a histone demethylase is inhibited. And it's been a pretty wild journey, as you can imagine, going from trying to understand how histomethylation functions in sort of transgenerational phenotypes, just as a very basic mechanism, to what we think is really major, clinical insight into the mechanism of Alzheimer's. So that's the conclusion, and I can tell you a little bit about the journey.
Yeah, definitely. I want to hear how you get from, A to Z. You're starting to look at C. elegans as, more of that simple model that people can refer to in a lot of those kind of initial preliminary studies or pilot studies. And now making this great statement as, hey, you believe that Alzheimer's disease is an epigenetic disease resulting from a loss of cell fate. So tell me how you got there.
So my lab actually, there's an intermediate step, which is my lab also studies mouse. And so, we actually found some initial phenotypes in C. elegans that suggested that histomethylation could be inherited transgenerationally and actually change phenotypes. And so the C. elegans is really, really good for that because the generation time is basically 3 days. So you can have multiple generations. It's a clonal, it's a hermaphrodite, so you can clone out, you don't have to mate. There's a lot of advantages for studying this. But when we found that, I was really interested in whether or not that actually might be true in mammals and whether or not there are inherited phenotypes due to inherited histomethylation. So my lab works in mice, and we went in to try and understand whether or not we are particularly interested in an enzyme LSD1, which is histone demethylase. It erases this H3K4 methylation, which is a histone modification, which is associated with sort of active transcription. And we think that it needs to be reprogrammed or erased between generations, because if it's not, it can sort of maintain transcriptional states. And so we set out, we originally found that in C. elegans, and we set out to see if that was true in mouse. And so basically the very simple experiment that we wanted to do was basically delete this enzyme in the germline of mice and ask whether or not you could get inherited phenotypes from one generation to the next. Mm-hmm. We really wanted to do that maternally in female mice. But right away, when we started to do those experiments, we realized that this histone demethylase, if you go to down the evolutionary train, basically, if you look at even down to plants or yeast or certainly Drosophila, it really is primarily involved in the germline. But right away in mammals and in mice, we saw that it was widely expressed in lots of different places, including it was not just in the female germline, but it was also in the male germline. Mm-hmm. So we had the opportunity to look in the male germline, and we did that. We actually very early on discovered that this histone demethylase is involved in stem cells. It turns out from many, many labs that this histone demethylase is required basically for all stem cells to differentiate. So the population that we happen to be looking at were testis stem cells, which go on to make sperm.
Got it.
the coincidence happens because we were trying to engineer a mouse in which we could specifically delete this histone demethylase, LSD1. It's also known as KDM1A in mammals, in the male germline, in the stem cells, and see what happens. And we didn't have the ability to do that. So basically, you do that through a technology called Cre-Lox, and you have to basically have the ability to have something that's expressed specifically in that cell type. We didn't have something that we could turn on specifically in the testis. So basically, the long and short of it is, instead of actually just deleting it, engineering it to delete just in a male mouse in the testis, we actually engineered it where we could delete it everywhere. So we could turn it off in an entire mouse, and then we could actually just analyze what happens in the testis and find out what happens to those stem cells. So we were actually taking an adult mouse and we were deleting the gene and then asking what happens in the testis. But of course, we were deleting it everywhere. And so when we did that, we got the huge surprise that within a series of weeks, basically all of the neurons in the hippocampus and cortex all die, and the mice become paralyzed.
Wow.
So that was not at all what we expected. It was not at all what we were studying, and it was a big surprise.
Yes. What'd you do at that point? Sorry, I have to cut in there. What were your first initial thoughts? And well, I want to get this answer, and then I want to break down everything you just said for the audience just a little bit further. But kind of what was that initial reaction?
Stunned. So, right away, I mean, it immediately suggested that you need this particular enzyme in order to maintain basically the entire structure of the brain. And that was stunning. Not at all what I expected, because I expected it really potentially, based on everything we knew, that if cells were dividing and changing what their fate was, like in a stem cell situation, you need LSD1, this histone demethylase. But in the brain, you have neurons, right, they're not dividing, they're not changing their transcription. And so it was completely sort of opposite of what anybody in the field knew about this enzyme.
Absolutely. So, no, thank you for beautifully explaining that research. And that's actually how I first really found you, was this work in epigenetics and Alzheimer's. And, you have a couple papers as well where you kind of talk about neuroepigenetic mechanisms in disease. So really excited to dive into this a little bit further. Now, I want to back up. My audience is definitely familiar with epigenetics. If you're listening to this podcast, you should be by now. But more specifically, DNA methylation as kind of that epigenetic modification. So when you're talking about, histone demethylase, can you just explain that and relate that to epigenetics? And then we'll talk about the LSD1 enzyme.
So basically, whenever you're talking about epigenetics, including DNA methylation, you're talking about changes that are not occurring on the DNA sequence, but that actually can affect whether genes are on or off. So a lot of times DNA methylation can do that directly, but in the case of histone methylation, so DNA is wrapped around basically like you would wrap yarn around a spool or a thread, and it's basically packaged into what's called chromatin, right? And so basically you take a huge amount of DNA and you shove it into this tiny little nucleus. And to do that, you can imagine that once you do that, it makes it very, very hard for machinery to get in and turn on genes. So what happens is, if you actually modify the packaging, which is the histones, then it actually changes the structure to sort of more open or accessible or more closed. And that basically helps facilitate whether genes are on or off. So histone methylation, basically, the series of modifications that occur to the packaging wherever that particular gene is can actually have a big influence on whether or not the gene is expressed or not.
So it's just everyone listening again to reemphasize that it's just going to be a different epigenetic modification, one that we don't hear about as often. in regards to everything I talked to, but one that is obviously equally as important, which is why we're discussing that today. So, this podcast episode is going to be a little bit more mechanistic based, but I want everyone to stick with me here. We're going to keep talking about these connections for epigenetics, histone acetylation, and how that interplays with DNA methylation and just epigenetics generally itself. We're also going to get into some testing methodologies, how this will hopefully be commercialized or the applications, and it'll be to scale, kind of like the DNA methylation is now. So, Dr. Katz, going back to that LSD1 enzyme, what did we know about it before? Did we know anything about it before you started kind of taking a deep dive? And then why did you choose that one as well to knock out?
So one thing I should add is, so if the audience is more familiar with DNA methylation, so the enzyme actually that adds DNA methylation is a complex called DNMT3A and 3B. And that actually, there's a direct, basically interaction between histone methylation and DNA methylation, because that actually complex actually lands on the packaging in order to be able to methylate DNA. So if the histone, which is the packaging, is actually methylated at K4, it will actually block the DNA methyltransferase from landing there. So when you have histone methylation, it blocks the DNA methylation directly. So there's actually an interplay between the histone methylation and the DNA methylation in terms of epigenetics. So that's one thing.
Perfect.
As far as LSD1, when we started actually down this road in mammals, there was only one thing that had been known, which is that it was absolutely required. So there was a mouse knockout made where they deleted the gene, and that dies embryonically. So that just says that it's required. That was the only thing that was known. And the reason why we got into it actually goes back to a very, very specific thing in C. elegans. There's a particular moment where the two cells that are going to go on to make the sperm and the egg, they actually lose this particular modification. And so we wanted to know, was this enzyme the enzyme that was doing that? The answer that turned out to be no. So, I did a very simple experiment back when I was a postdoc, basically in C. elegans, asking whether this histone demethylase was demethylating this particular histone modification at this chemo. The answer was no. But actually what was interesting at the time is, and what got me into this whole thing, is that when you deleted LSD1 in worms, what happened is the population of worms actually got more and more sterile over generations. And that first got me interested in the idea that, well, maybe the actual histomethylation is changing over generations and is actually causing the phenotype. So that's where it all started.
Yeah, very interesting. No, I like to always hear how these experiments get started. And it seems like you had a lot of questions beforehand where, you kept getting the answer, no. Okay, let's try this. So
it just kind of a long way from where we ended.
Yeah. Just kind of the nature of research, right? Yeah. So we've done this. You've knocked out the LSD1 enzyme and C. elegans. You said, okay, maybe as a result over time, those models ended up becoming more sterile. And then you're knocking it out in mice, and then we're seeing massive neurodegeneration, paralysis. Hopefully this is showing it's leading and playing a role in Alzheimer's disease. So how do you make that connection? If you knock out this enzyme, you see these mice, how are you then taking those mice and connecting that to Alzheimer's disease?
Yeah, I mean, to be honest, so I get this question all the time. Why, why did you think that LSD1 was involved in Alzheimer's disease? And the answer is I never did. Not a million years, including even after we actually we engineered this mouse where we got rid of LSD1 in an adult mouse and we saw that it got massive neurodegeneration. I still had no inclination whatsoever that it was going to be involved in Alzheimer's disease or any neurodegeneration at all. But it was just simply too interesting not to follow up. So basically, the way we first got to Alzheimer's disease, so this histone demethylase, it erases a histone modification, which is associated with actively transcribed genes. So it's a repressor. It turns genes off. So we simply did a basically experiment. We said, all right, in all of these neurons that are dying in the brain, what are the genes that turn on when you lose it? Well, the idea was, could we identify some of the pathways that might be between, losing LSD1 and causing neurodegeneration? So we identified genome-wide in the entire genome, what are the genes that activate when you lose this enzyme? And, as you do when you do this sort of genomic experiment, you simply say, all right, well, what kind of pathways are affected? Or does this look like any other experiments that people have done in the past? And when we started to basically search in the computer and look for this, what it turned out is that the genes that turn on when you lose LSD1 in a mouse look very, very similar to the genes that turn on in human Alzheimer's patients.
Okay.
So that was the first time that we had any inclination that this could be involved in a human disease at all.
Yeah. And did you mention 2 points, Dr. Katz? Did you say there was another point as well?
No, that was the
main one,
right?
Okay.
We just wanted to know why, when you lose this enzyme, why do neurons die? And it turns out that the genes that turn on when you lose these enzymes are really very, very similar to the genes that turn on inside the brains of Alzheimer's patients.
Gotcha. And did you say you did that through a GWAS, a genome-wide association study? Or you were just looking at it?
It was a simple RNA-seq experiment. So we basically identified gene expression changes genome-wide, and then we just compared it using, initially just gene ontology, which is just a fancy way of saying what pathways are affected, and basically through the computer you can ask, in any experiment that people have looked at gene expression changes, do they match what you see? And then basically the computer can say, yes, this looks very similar to what was seen. And it turns out that, large consortiums, we're starting to say, all right, are there any important gene expression changes that are occurring in Alzheimer's, patients? So they had collected hundreds of specimens from deceased Alzheimer's patients who had donated their brains. And they had asked, all right, what are the genes that turn on in these Alzheimer's patients? And they had published a huge set of genes. And the interesting thing about it is the genes that turn on in an Alzheimer's patients don't make any sense to anyone. There's all kinds of different pathways, and people argue about, is it one or is it the other? Is it all of them that's causing the disease? And so actually for the Alzheimer's patients, or for the Alzheimer's researchers, it didn't actually help that much to know, because there's so many different things.
Yeah. No. Super interesting as well. I don't think I mentioned this on the podcast before, but Alzheimer's dementia definitely runs in my family. I've had family members, lost due to the disease, and it's just heartbreaking. So this is super interesting to me from more of a personal level too. I know I have the APO34 variant, and I'm also the type of person who wants to know everything about myself. So if there's anything that I can do to map my epigenetics, to map my genetics, understand my disease risk, how I can, hopefully decrease that risk too, I'm definitely all about it. So
Alzheimer's itself is actually interesting, because people have looked very hard for strong genetic correlations. So there are two sets of genetic contributions to Alzheimer's disease. So there is a very, very rare set of patients that have mutations, and if you have those mutations, you're guaranteed to get Alzheimer's disease, familial forms, in your very, very young 30s and 40s. This is exceedingly rare. And then the vast, vast majority of Alzheimer's patients, which is exceedingly prevalent, in the Western world and in the United States, are basically sporadic. So there's not a lot of evidence that there's much that's inherited at all. The only exception to that is what you were talking about. So ApoE is a particular gene, and there's a variant in it. If you have an ApoE4, if you have 2 copies of that in particular, even there, you're by no means guaranteed to get Alzheimer's. That's the only thing that will substantially increase your risk. Those patients tend to have not just a family history, but a family history where people got it particularly early. So most people get Alzheimer's disease, something like 75, 80, 85. And sometimes if you have a large number of people in your family who've gotten it a little earlier, like 60, 65, then sometimes you can be carrying that particular variant. You can get tested for that. On 23andMe or any of the other tests.
Yeah.
Again, that's very rare. Even those alleles, those particular variants of those genes are very, very rare. So the vast majority of time, even if both of your parents had Alzheimer's disease, you're really not much above the average risk in the population.
Well, that's good to know. That's good to know. That'll make me sleep better tonight. I know that's for sure. So yeah, no, Dr. Katz, again, a wealth of knowledge. Really, I love your story from beginning to end, and it's definitely not the end quite yet. We're still learning what this all means. So, before we hopped on, we were chatting a little bit about how we best wanted to format this for the listeners, and you mentioned that a lot of epigeneticists are very supportive of your work. Yes, green light. This looks great, but you said more people within just like kind of the Alzheimer's, I guess, research community are a little bit more hesitant or skeptical. Can you maybe explain why? And what's the evidence? What are people asking? What are people wanting to know here?
Yeah, so I think that the reason why the Alzheimer's field is so skeptical is that there's been a huge history of people coming in and saying, "Oh, Alzheimer's disease is really this or really that," and we've been studying. Alzheimer himself identified the very first misfolded proteins in the brains of human Alzheimer's patient. That was over 100 years ago, right? So we've been studying this disease for a long time, right? And you would think with all the number of people and now the huge amount of resources going into it that we would have a lot of insight, and we have learned a lot. I'm not going to downplay that at all, but there's a history of people coming in and saying Alzheimer's is really this, and it's really simple, and it can be cured or whatever, and those things have not turned out to be true. So I think people are pretty right to be skeptical. That being said, I think one of the things that's not so great is I think as scientists, you want to be skeptical always. And we were very skeptical ourselves. And then you want to see whether or not the evidence actually can change your mind, right? So once you have a certain number of pieces of evidence, and I can go through what we have now subsequently been able to show, then I think you start to change your mind, right? And I think the epigenetics community has been more open to hearing, look, you've got all this evidence that suggests that this enzyme could be involved. Whereas the Alzheimer's community, I think, is a little more, well, we've seen it all before. And so nothing can really be true, right?
Right. I think they don't want to get their hopes up, right? We've seen a lot of those. I don't know, even if you want to talk about this as well, and you'll be able to do this better than myself, but some of those falsified, images with the Alzheimer's and, thinking we've made a really big breakthrough. You'll see kind of, I'll see at least in my Google alerts or in the news, hey, something new, or, we've found a partial cure or something, right? And then something else happens. So, tell me what else people are saying. And then I do want to hear, again, your evidence for to support this.
Yeah, I mean, even currently, right? So there are 2 drugs that have been approved very, very recently. And there's a lot of controversy surrounding particularly the first drug, but even both drugs, because the second drug that looks a little bit more promising, even there, to say that it really is an Alzheimer's drug is, I think, maybe overreaching a little bit, because it doesn't modify the progression of the disease at all, right? So the people, even if you take the drug, and even if you say it works in the trials, that the people still get degeneration, they still progress the same. What it does is that it slightly modifies symptoms in the very early phases of the disease. So, and it's questionable, in my opinion anyway, as to how much you would really notice the amount that it modifies it. Obviously, if you give people cognitive tests, you can test it, and it's statistically significant. And I don't want to downplay that at all. It is our first drug that does anything to the disease. So that's a fantastic thing. But we are nowhere close to having anything that you would describe as a cure, because none of these things none of the drugs even claim to modify the progression of the disease. So
Got it.
If you have Alzheimer's, what you don't want to do is have your dementia progress and lose all of the things that are associated with those patients, which is starting with short-term memory and then leading to, really major cognitive defects. So, as far as what we found is, so once we found that the genes that are changing in our mice were very reminiscent of the genes that are changing in the human patients, the first thing that you might want to know is, all right, well, is there any evidence that LSD1 itself, this enzyme that we were studying, is affected in human Alzheimer's patients? So obviously a mouse is not a human, and so you don't want to make a claim about the disease in a human unless you have any evidence that it's affected. The interesting thing, so just to give your audience a little bit of context about human Alzheimer's disease, and
Yes,
I will join the lay people in saying that I really knew almost nothing about Alzheimer's when we started this. So I've come a long way, obviously, but I knew very little. So there are two different proteins which misfold in the brains of Alzheimer's patients. So they are called beta amyloid and tau. And actually, in the very original drawings from Alzheimer's himself and the very first patient, you can actually see that he drew these. So what happens is you can see in these patients that there are proteins, which, for the lay audience is basically the building block of the cell. Everything is made out of proteins. So they normally have a particular structure. And what happens in the case of these two particular proteins only in Alzheimer's patients is that they misfold so that they lack their normal structure. Okay. And so those two things we've known for a fairly long time correlate with the disease. So you tend to see them inside the patient's brains who have Alzheimer's disease. We now know, for a long time, there's been a lot of people focused on beta amyloid because, and quite rightly so, because the beta amyloid, hypothesis, if you will, came about when, first of all, people identified that one of these proteins was beta amyloid. And then very quickly thereafter, they showed that if you have familial mutations that affect beta amyloid, that those people get Alzheimer's guaranteed in their 30s and 40s. These are the very rare patients that have mutations. So that was pretty good evidence, right? So you have the patients have the misfolded protein, and if you have the mutation that's in the pathway, you get Alzheimer's disease. So you say, look, finished, done, right? That's got to be the disease, right? Made a lot of sense.
Just one part. It's just one part, though, right? Is what you're getting at here.
It turns out that there's another misfolded protein, tau, and one of the things that happened right away that should have been maybe a clue is that there's a lot of people that have misfolded beta amyloid that don't have Alzheimer's disease. So there is an overall correlation, but the correlation is not by any means perfect. Now, as we've gone on further, there is a better correlation, much better correlation between misfolded tau protein. And so now there's sort of a revised beta amyloid hypothesis that basically you start to get misfolded one protein, beta amyloid. Later on, you get misfolded tau, and that eventually one or both of those proteins, probably tau, but maybe some contribution of beta amyloid, are actually interfering with something that the neuron needs to do and causing the neurons to die. So what really happens in Alzheimer's disease is you get millions and millions of neurons that die. Okay. And that's why you get dementia.
Gotcha.
So the big question is, how in the world does tau, for example, cause a neuron to die?
Yeah. And that's what you found a little bit with your study. Is that correct? Yeah. Right?
So the interesting thing is, going back to what I said, is so as soon as we found this, we said, all right, is there any evidence that this histone demethylase is affected in human Alzheimer's patients? So we went in and we looked at this this particular protein LSD1 in Alzheimer's patients. And one of
Yes.
the things that's been really frustrating for the field is that even though like tau is supposed to be killing neurons and causing probably Alzheimer's disease, right? People have said, all right, it's interfering with things inside the cell. So what are the other things that are basically getting stuck when these proteins misfold? So people have
Okay.
looked very, very hard for other proteins or other pathways that could be affected. And basically it's been exceedingly frustrating. Nobody has basically found it much of anything. There are a few
Yeah.
things that have been found, but not a lot. So we were, again, floored because we went and looked at this particular histone demethylase, and it's normally in the nucleus because it has to regulate genes. And the buildup of this misfolded tau actually happens basically in the cytoplasm. So they're in different locations. But the moment we looked inside the human Alzheimer's brains, we found that this histone demethylase was basically perfectly localizing to misfolded tau.
Gotcha. Okay, perfect. So oh, that is exciting. I can't imagine. So you found this pathway, Dr. Katz, and then you were super surprised kind of with these findings and relating it to Alzheimer's disease. So when did you actually make the discovery with the tau protein?
So that happened at, it's funny because we had the original, this mouse project, and we showed that you get neurodegeneration, and we wanted to publish that. And actually people said, oh, you should look to see what's happening in the human patients. And I said, well, that's going to take us, years. I knew a little bit, and digging into literature even a little bit, it was very difficult for people to find things. So I was very hesitant to do it, but I got pushed I happened to be at Emory University. We have a very strong Alzheimer's group. We actually have, for the whole southeast of the United States, one of the only brain banks where people can donate their brains if they have Alzheimer's disease. So we had access to the patient samples.
Gotcha.
And so it wasn't that actually hard to take a look at this. And so it turned out I got pushed to do it, and we did it. And I was shocked because it was so obvious. So basically what happens in Alzheimer's disease cases, it's called. So once the people die, they donate their brain, and then they do pathology, which is basically an autopsy. And they ask, do you have this misfolded tau in your brain? And mostly, do you have misfolded beta amyloid in your brain? So technically, you can't be diagnosed with Alzheimer's until after you die, because the only way we can really prove that you had it is to look inside the brain and look at these. So people get diagnosed with it, but actually to prove it and to be considered a true Alzheimer's patient, then you actually have to donate your brain and be diagnosed that way. So in any case, what's interesting is, so you diagnose this by looking at this tau protein. And it turns out that if you use the same exact technique and you look at LSD1 instead of tau, then you can take the people, the pathologists who are the doctors who actually diagnose this, and they can't tell the difference. So they look exactly the same, which was a huge surprise.
The LSD1 and the tau protein, you're saying?
Yes.
Yeah. Yeah. And you know what? That's great that you have that kind of brain bank, if you will, at Emory. I think the University of Kentucky here does that as well. I know we have some really good Alzheimer's research going on.
Fantastic Alzheimer's group. And they also have a brain bank for sure. Yep.
Yeah. So, I mean, that's great. And what year was that, or when was that when you started notifying that kind of in the human brain as well? Because that's my next question is, hey, how do we take this from the C. elegans to the mouse to the human? What's the translation to the humans? What's that relationship? But it seems pretty strong.
We first made the discovery, the first time we saw that the mouse was getting neurodegeneration was basically in 2013.
Okay.
We published our first paper, I think, in maybe 2017, something like that. Showing the first, and that included the first evidence that it was actually could be affected in the human patients. And, now we're in 2023, and we have substantially more evidence that I'll talk about. But so it's taken, maybe 10 years from the very, very first discovery. Yeah.
No, that's great. I was just kind of, I would say that's not that long considering everything, right? It actually moved pretty quick because I think once you kind of find that little, golden nugget, if you want to call it that, and then kind of take it and run with it. Like you said, when you looked at those human brains, you saw you can tell the difference between the LSD one and the tau. So this is great. And do you think that this could act as a way to diagnose Alzheimer's in patients beforehand? Like, how are they, doing these clinical trials with all of these drugs if you can't technically diagnose Alzheimer's, when they're still alive? Or that's hard.
So we now in the last just couple of years have figured out way through scanning to actually, you can identify the buildup of beta amyloid and tau in a human brain in a live patient.
Got it.
So that can actually be done ahead of time now. People don't do it very often because you have to take a compound and insert it into your bloodstream. It goes everywhere, and then they do imaging. It's not an easy procedure. So it's not done that often, but it can be done. So as far as, for a lot of these experiments and things are the way people have done it, it's actually, for example, the clinical trials for these drugs that have just come out, it's actually interesting because, the inclusion criteria are difficult because you get included based on the dementia, which is basically you say you're having short-term memory issues. But the good clinicians very easily can tell the difference between, for example, Alzheimer's and, let's say, frontotemporal dementia, which is the second most common form. But it's not perfect, actually. And it relies on and sometimes they're wrong, right? So there are people maybe in those clinical trials, it's possible you could have a small number of people who actually don't have Alzheimer's.
I think so. I think that just can be hard, but hopefully with this new imaging, and then we'll have better stuff in the future, I'm sure, as well. So did you mention that you had another piece of evidence you wanted to talk about, Dr. Katz?
Yeah. So so the first two pieces of evidence said, all right, in the human patients, it's a correlation, which just says, all right, we know that there is this protein which misfolds, which people think might cause Alzheimer's. We know that this histone demethylase is there where it's not supposed to be. And so then it means it could be affected. It doesn't say that it is, it says it just could be affected. And then we know in a mouse model that if we actually just get rid of this enzyme, we can cause neurodegeneration. So that's causative, right? Those are two pieces of evidence. But the big missing piece is, all right, there's no connection. Is there any evidence that actually tau can truly block LSD1, that it can inhibit LSD1? Right. So basically, so the model is, LSD1 is a protein which regulates genes in the nucleus, and tau is a protein which misfolds in the cytoplasm of these neurons. And the model is a pretty simple one, which is to say that this enzyme LSD1 is supposed to be in the nucleus. It's getting stuck in tau. It can't get in the nucleus where it can't repress genes. And that's causing all of these different, pathways to activate. That's the model. So the question is, that model makes a couple of different predictions, right? So it suggests that if the main way that tau is actually causing problems is by inhibiting this enzyme, that you should be able to manipulate that, right? So you should, first of all, you should be able to show that tau can maybe prevent LSD1 from being in the nucleus. And you should be able to say, all right, if that's true, if you were to reduce LSD1, let's say in a mouse model, it should make everything much worse, right? Because it should be much easier for LSD1 to get stuck. And if you were to actually increase the amount of LSD1 in a tau mouse model, then potentially, even if some got stuck, some would still get to the nucleus, and it might actually, help. So that's what we set out to do. We took a tau Alzheimer's. Well, I shouldn't actually call it an Alzheimer's mouse. It's complicated because the tau mutation actually came from a frontotemporal dementia patient. So it's really a tau model, tau mouse model. But anyway, so if you take a tau mouse model and you say, all right, if the prediction should be that if you reduce LSD1 in that mouse model, it should make everything much worse. And if you increase the amount of LSD1, it actually could potentially make things better. And so the tau model, we should say, is a little bit, I don't know if controversial is the right word, but the tau mice never get nearly as severe as human patients. And it's not really known why. Maybe because the mice don't live as long as a human patient, so it takes time. But in any case, in the tau mouse model, basically, they've been engineered for tau, this protein, to misfold, and they do get some neurodegeneration in those mice.
Okay, so I'm following. I'm taking a lot of notes, but I'm following here. And I guess to cut in, Dr. Katz, point being is you can't knock out the LSD1 in humans. That's not possible, right? So you're doing kind of, you're taking the Alzheimer's model from a human and trying to replicate that in the mouse by using that tau protein, and we're just not maybe getting as a clear picture as we want quite yet to scale.
So that's what the tau model is. So what we did is we simply took the tau mouse model, and we said, first of all, can tau actually block LSD1 from being in the nucleus? That turns out to be completely true. So if you look in the tau mouse model, LSD1, which is supposed to be in the nucleus, it now is completely gone from the nucleus in those key neurons. So that was great. And so now it's no longer just a correlation, but we actually show that tau can inhibit LSD1. And then we said, all right, if we reduce LSD1 in the tau mouse model, it should get much worse. That turns out to be very true. So what happens in the tau mouse model is tau misfolds over 6 to 9 months, and then they get neurodegeneration around a year. It's not so severe, as I said. But if you reduce LSD1 just a little bit, even by 30%, those mice, once the tau starts to misfold, they get a way more severe version of neurodegeneration that actually looks a lot like the human patients now for the first time. So it makes things much, much worse.
Okay. Well, and if you increase the LSD1, are we in the nucleus for tau? Are we seeing those mice get better?
Yeah. So we did that, and we did that in a particularly, I don't know what the right word is, but maybe an aggressive way, which is we said, look, so there are people who have been able to modify the tau mouse model so they get less neurodegeneration, but those are typically modifications from birth. So genetic modifications and things. We decided, look, if we're right and LSD1 really is the thing that's causing neurodegeneration and by tau and in the human patients, then we should be able to increase tau not just from birth, but right after the tau, increase LSD1, not just from birth, but right after tau has already misfolded. Basically, the equivalent to the time that you would walk into the doctor's office as a patient. And that should actually have an effect. So we did this in a fancy way. We engineered the mice basically so that we could actually increase LSD1 in the tau mouse model after they already have tau that's misfolded just before the neurons start to die. And we were able, for the first time, to block neurons from dying.
Oh, wow. Wow. Awesome. That's the $1 million question then, right? Can tau inhibit LSD1?
Yep.
If we see it go up, do we get better? If we see it go down, do we get worse? And again, I would call myself definitely more of an epigeneticist than an Alzheimer's researcher, but the evidence seems, pretty strong for me at least. So where do we go from there, Dr. Katz? How does this work? Advanced drug discovery in Alzheimer's disease. I guess, what are you working on now? It seems like you've proven kind of all three of those points in a mouse model. Again, I don't know how much more you can do or show. Do you move to a human model? How does
it's difficult. Once you get to a certain amount of evidence that there is not a lot left to show except for whether or not doing the same thing in a human can actually block neurodegeneration, right? And to do that, you actually need a drug. You can't engineer the humans like you can. Right. So we are very interested in identifying a drug. We are currently starting on a drug screen to basically find this. So again, the model is a pretty simple one, which is we think that tau, when it's misfolding, is blocking this enzyme that has to be in the nucleus repressing genes, and it's blocking it. It's getting stuck. So it's not there in the nucleus, and lots of genes are turning on. It's interesting because it's from an epigenetic standpoint in that, and we can get back to this if you want in a minute, So what are the genes that turn on? Why is it all these weird pathways? Because it's a whole bunch of weird genes. It almost looks like the set of genes that are easiest to turn on in a neuron that this is normally blocking, and it's a bunch of very strange genes, but it causes a number of different pathways to turn on, and those are all the same pathways that you see in the human patients, and those pathways, a number of which are probably all killing the neuron.
Got it.
So we just think that it's an intermediate between tau misfolding and the downstream pathways that are killing a neuron. Right now, the sort of a consensus is that, maybe tau is somehow inhibiting all of those pathways. We think that it's really inhibiting a single target. That target is normally affecting lots of pathways. And so that's how you get all those pathways affected.
Yeah, no, I mean, this makes sense. I was so excited to talk to you, but I think a little bit nervous to see, if my audience could follow along here, but I'm following very, very well. So we have this tau protein. It's going to be one of the misfolded proteins that happens in Alzheimer's. So when this happens, we do see, I said, tau inhibit the LSD1. We need the LSD1 to help regulate our gene expression. But when tau has an effect on this LSD1, it's actually turning on a lot of genes that shouldn't be turned on. Thus, Alzheimer's happens. Would that be a good summary?
Yeah, that's it.
Of course, I'm taking notes here and trying to walk through it myself. But this is seriously groundbreaking work, and very, very excited to follow along. So, is that then again your main priority right now is to look for drugs that are helping increase the LSD1 or something to work on that pathway?
We have. That's the main priority, yes. The only other remaining question that we have is, why does LSD1 get stuck in tau when lots of other proteins don't? That's a big question, and we have some evidence for that. It gets pretty technical about what we're talking about.
Yeah.
about that, but that actually is not necessary to know that it's involved in Alzheimer's disease. But I think it is an important part of when we're evaluating the drugs that come out of our drug screen. So what we're looking for is a drug that can block this interaction between tau and LSD1 and allow LSD1 to still go into the nucleus to repress genes even when tau is misfolding.
Got it. Yeah. And again, to all my listeners, this is really, really important. A lot of times, we do spend a lot of time on epigenetics and longevity and aging and correlation, which is not causation. But here we're having both, right? We're having, tau misfolded protein equals an effect on the LSD1. So that is the correlation. The causation is, if we actually knock out this LSD1 in mice, they actually tend to have worse outcomes too. So this is, I know it's more mechanistic. I know it may be, again, a little bit harder to follow, but this is where we can actually then go into this drug discovery pathway and bring this to application and bring this to market. So just super, super fascinating. I know we're getting close to the end here. I feel like I could talk to you all day, Dr. Katz. I want to give you a second to talk about a program that you've established with the nearby liberal arts college. I think it's Oglethorpe, if I'm correct, called CURE. So would love for you to be able to promote that and even talk about how it helps promoting DEI in science.
Yeah. So, going back to the beginning of this project, right? So we started this by doing very basic experiments on a tiny little microscopic worm and trying to understand transgenerational effects, things that are passed on from parents to the offspring. And what's really great about this model system, C. elegans, is that it's very quick and easy to learn, right? So there's a new generation every 3 days. They're very easy to take care of. They don't cause any there's no health problems. They're found commonly everywhere in the soil. They're in rotting fruit, everything. So that allows us to actually do something that I really wanted to do for a long time, which is to try to promote science. One of the big problems in science is that there's sort of an accessibility problem, right? So in order to become a scientist, a lot of people have to go to pretty fancy universities that have a lot of money to put into this. And then on top of all of that, they have to really opt in. So you have to be the kind of person who's willing to go approach a professor like me and say, look, I want to learn how to do this. And for a lot of people, that's just not realistic, right?
Mm-hmm.
So what we're doing is we're missing out on a huge population of people who could be fantastic scientists. So what we wanted to do was sort of bring science, instead of an opt-in model, basically just a system where people would be exposed to doing research without having to opt in. So we went to a small liberal arts college here in Atlanta that has a huge underrepresented minority population. Many people are the first in their family to go to college. And what we did, it's a tiny liberal arts college, so they don't have any research whatsoever, no facilities. But as I said, this kind of research doesn't require a lot. So we started this actually with very little. We basically said, all right, we're going to integrate short research experiences into the entire curriculum. Right. Mm-hmm. So this is what CURE stands for. CURE is not specific to our program. Ours is, we technically call it a pipeline CURE because it happens over all of the time of college, all the different years, freshman through senior. But CURE just stands for course embedded research experience. Right. So anytime you take research and put it into the classroom in the context of a course, that's called a CURE. So what we did is we said, all right, we're going to try to go beyond that, and we're going to slowly introduce this system into early classes. So the students there start to take, small research experiences, 2 weeks, just like you would have a lab in a class, and they learn the system. And then as they go along, by the time they get to a junior or senior, they can opt into much more advanced, research programs where over the entire semester, they're actually doing experiments. And it's actually some of these experiments have resulted in some really amazing epigenetic results where the students themselves have found really elegant examples of transgenerational effects where you can actually have traits that are passed on from one offspring to the next.
Yeah. No, that's great. And I meant to even say this earlier as well, for anyone who's like, what's the germline? What's transgenerational epigenetic inheritance, which is a mouthful. I did talk about that topic very specifically with Dr. Michael Skinner in one of my last podcasts from Washington University that you can watch. And then I also talk about, I spoke with the, bringing DEI into science with actually Dr. Kristen Bird from Ohio State. And I just think it's so important. I think the more we talk about it, the more we can get people involved, because you're right. I remember being a freshman in college and being scared to go to professors' doors and ask for, research positions that, have to start off early in your career, or, you have to get in, and then that's when you really get into some of the deep science like you're doing now. So, very much a big thank you, I'm sure, to everyone that you've helped there in that CURE program. And I'm excited to see where that goes and how it helps promote, your epigenetics and even the work you're doing.
Yeah. One of the things that's really hard is that, if you don't have a scientific background, you may be a biology major in college. By the time you realize what research is, maybe it's your junior or senior year. A lot of people have already been doing research before that. You don't know where to start. So what happens in this case is because they're just doing it. So in order to, people say, how do I get into this program? All the freshmen come and ask there, how do I get into this program? And I said, there is no program. It's just a biology major, right? So any student who enrolls as a biology major gets this, just whether they want to or not, they get the experience. They don't have to opt into the research at the end, but they all get trained. So the point when they're juniors and seniors, all they have to do is enroll in the next class and they already have the training and background. So there's no opt-in here, right? No realization that they have to do it and go get the training on their own. They all already have it. And what that means is that it sort of lowers the barrier to entry because you don't have to know that you're entering the program until you find out, oh, I really like this.
Yeah. I mean, that experience is essential. So just, supplying it with people, whether they want it or not, they can choose to, leave it as is. But, that is definitely needed and such a competitive atmosphere too, especially if you're not getting any, of that, kind of experience this, from high school, which is very, very rare. So, excited to see that grow, Dr. Katz, and hopefully be offered, all around the U.S. or all around the state someday.
We have, for the first time, this university is very old, 150 years, 180 years, something like that. And basically, as far as we know, they've never had students go on to be a PhD or become a scientist. And we've now had 2 go directly to PhD programs. So,
oh, very cool.
Yeah, we're very excited about it.
Yeah, and we'll only see that number increase as well. So, Dr. Katz, I have one last question for you. Sure. It has nothing to do with anything we've talked about so far. I ask all my guests this. If you could be any animal in the world, what would you be and why?
An animal?
Yes.
Oh, my.
It's a heavy loaded question.
Yeah. I don't know. I love penguins. They're very, they're very cute. And, the idea of living in a, particularly, like Antarctic penguins that live in these really, really cold climates and have no problem with that. I think that's pretty amazing.
One thing, no one's ever said penguin. So have you ever seen one like in a natural landscape?
Not so much, but I'm going on a trip to South Africa to see colonies of penguins. So, in a couple of months, be able to see some, which will be fantastic.
Please send me pictures. I bet that will be beautiful. I bet it will be breathtaking too. Well, this is great, Dr. Katz. For anyone who's interested, I don't know, maybe they want to reach out about the Cure kind of offering, or, learn a little bit more about how to get involved in science or just your work in general. Where can they reach you?
You can, easy thing is to find me on Twitter at Katz_lab, or you can reach out to me on my email, which is djkatz@emory.edu, or the website if you just Google search David Katz, Emory University, you can find our lab website.
Awesome. And I'll make sure to put all those links for those listeners out there who are interested. So thanks everyone for listening and keeping up with Dr. Katz and I at the Everything Epigenetics podcast. Remember, you have control over your DNA. So tune in next time to learn how. Thanks, Dr. Katz.
Thank you.